Hello, everyone. Good afternoon. Welcome to this second edition of our EMBRC Technology Webinar series. This is a space to inspire collaboration and cutting-edge research. While people are still joining, I would like to share a little introduction of our organization, EMBRC, for those who are not familiar or are new to our community. So we will put the link to our website in our chat. So the European Marine Biological Resource Center is a research infrastructure. It connects more than 80 marine stations, universities, and research institutes in 10 countries in Europe. We provide expertise for scientific and applied research and access to technology platforms, to marine organisms, to ecosystems, and also to data. We support a wide variety of users coming from the private sector and also from academia, and we touch topics such as environmental monitoring, marine biotechnology, marine conservation and ecology, developmental biology and evolution, drug discovery, and many others. With these webinars, we want to promote knowledge transfer and innovation, and we're very excited to bring together experts, researchers, and enthusiasts to explore the latest technologies and challenges in emerging fields, such as marine observation and marine aquaculture, like today, marine biotechnology. And we want to welcome very warmly all the people who are joining us and following this webinar from other continents besides Europe, such as Asia, America, and Africa. I just want to remind you that webinars will be presented in pairs for each series, and it will include a speaker that is working in a research lab from academia, from one of our partners doing applied research. And the second webinar is a company who will be sharing its expertise. So as you can imagine, with this huge network of more than 80 partners, EMBRC supports a range of applications. So our technology webinars will be changing over the time. This webinar series is dedicated to marine aquaculture. This is a field in which Europe is a world leader in research and development, and for which EMBRC has facilities for performing things as simple as a feed trial or as complex as genetic studies for applied selective breeding, for environmental impact assessment, or if you want to test innovative disease control strategies. We can also serve users looking for side stream valorization or upscaling and many other applications. I encourage you to access our services using our transnational access programs, which allows you to travel and visit the facilities to perform your experiments, your proofs of concepts, and also discuss with experts in the technology of your choice. We have several European projects supporting this transnational access experience, and we will put in the chat the link where you can find all the calls that we have open now, so you can access our services for free. Well, now that all our participants are online, I will continue to introduce our speaker of the day. So we are honored to have Dr. Pantelis Katharios, the founder and CEO of Aquatic Biologicals. This is a spin-off company of the Hellenic Centre for Marine Research, which is an EMBRC partner. Pantelis, besides leading a company, he's recognized as the first, marine biotechnology company in Greece that is exclusively focused on health of aquaculture fish. He's a distinguished researcher interested in pathologies affecting marine cultured and ornamental fish. He has a particular focus on emerging diseases in novel aquaculture species. A significant aspect of Dr. Katharios' research involves understanding molecular mechanisms that determine the virulence of pathogenic microorganisms and their interactions with fish hosts. This includes exploring bacteriophages, as he will tell us, as alternative treatments in aquaculture, particularly in fish hatcheries, and developing autogenous vaccines to enhance fish health management. With over 75 research and articles in peer-reviewed journals, we are very honored to have today Dr. Katharios as a speaker today in our webinar. Thank you very much, Pantelis, for accepting the invitation. The floor is yours.
Thank you very much. The honor is mine. I would like to thank you and the EMBRC for inviting me to give this webinar. And I will start. I'm sharing the screen. So can you see the screen?
Yes, perfect.
Okay, so today I'm going to talk about innovative disease control strategies for marine aquaculture, and I'm going to present the work that we have been doing in HCMR. As Mery said, my principal job is in research. I'm in the Hellenic Centre for Marine Research, but I also lead this spin-off company, Aquatic Biologicals, that I'm going to discuss about the activities of the company as well. Aquaculture has been identified as a hotspot for the development of antimicrobial resistance. This concern is not just theoretical, it's increasingly being recognized across various domains. For example, as highlighted here in this slide in The Guardian, the combined challenge of climate crisis and excessive antibiotic use pose significant threats to the fish farming industry, potentially undermining its sustainability. Moreover, this is not only flagged by the media. At the European level, institutions like the EU Science Hub of the European Commission have raised alarms about antibiotics in aquatic environments and the associated risks of drug-resistant bacteria. Together, these points underline the urgency for addressing antimicrobial resistance also in aquaculture. This growing concern about antimicrobial resistance is particularly significant when we turn our attention to the European and the Mediterranean marine aquaculture. Marine fish aquaculture in Europe is dominated by salmon. Salmon aquaculture has always been the focus of the big pharmaceutical companies because of its value and market size. On the other hand, the non-salmon in marine aquaculture is underserved because it is relatively smaller compared to salmon. However, this market is now growing. In the Mediterranean, for example, we have reached the production of almost 500,000 tons of fish and 1 billion juveniles per year. One of the big differences, however, between these two markets, salmon and the Mediterranean aquaculture, is whereas in salmon aquaculture we grow just one fish species, Atlantic salmon, in the Mediterranean we grow approximately 20 different fish species. And it is this fragmentation of the market that is the biggest impediment for the big pharmaceutical companies to invest in developing and licensing new medicinal products, amongst which are the vaccines. So let me give you some figures to put this problem into context. 400 million euros is the loss of revenue in the Mediterranean aquaculture due to diseases. 4 is the number of diseases which are covered by licensed, authorized vaccines in the Mediterranean aquaculture of the 20 species, whereas in salmon aquaculture, if I'm not wrong, we have vaccines for 14 different diseases. And that leads us to the third number, which is the quantities of antibiotic used in the Mediterranean for every ton of biomass produced, which is 100, whereas in salmon aquaculture, this figure is much, much lower, it's 1 gram, because they have the tools to mitigate the problems of bacterial diseases. So what can we do about this? So the first thing is to find ways to bring more vaccines in the market. And the second is to find alternatives for antibiotics. And this is actually what I'm going to discuss with you this afternoon. I'm going to talk about autogenous vaccines for the first problem, and for phage therapy, which is a very good alternative for antibiotics. So let's start with the autogenous vaccines. Autogenous vaccines are vaccines which can be used only when licensed, authorized vaccines are not commercially available. So these are vaccines which are used for vaccines not covered by vaccines which are already in the circulation. Their application is exclusively in the farm where the pathogen was isolated, because they are also made from the same pathogen. So what you need to do in order to develop a autogenous vaccine, you need to be very sure that the disease is not covered by commercial vaccine. You need to go to the fish farm, isolate and characterize the pathogen, and using this pathogen, make a vaccine for that particular company. So their use is limited in terms of locality. They should be supervised by a veterinarian, they should be prescribed by a veterinarian, and there is no requirement for authorization. So this is more or less the definition of autogenous vaccines. In my group in HCMR, we have been working with autogenous vaccine development for some years now, and I will just show you some examples of autogenous vaccines to show you the efficacy of these products, because these are very, very efficacious vaccines. I'm going to talk about Aeromonas veroni, which is an emerging pathogen for the sea bass aquaculture in the Mediterranean, and Vibrio harvae. Vibrio harvae is a pathogen with global distribution, but so far we don't have any commercial product for this one. So in the pictures on the left, you can see a sea bass which is affected by Vibrio harvae, and on the right, a sea bass which is affected by Aeromonas veroni. So this is a vaccine that we made using Aeromonas veroni, and we have vaccinated small fish, juveniles of 3 grams in weight, using an immersion vaccination. The route of administration was through immersion, a bath. And the challenge is also by immersion. It's a very effective challenge test. So what you can see here in this graph is the mortalities of the control group, the non-vaccinated group, which almost reached 100%, whereas at the same time, the vaccinated fish, they have a survival of more than 90% and 85%. So this is in the index you see here, RPS. It's called relative percentage mortality, and it is an index that we are using in order to evaluate the efficacy of these vaccines. We have also used this vaccine as with the intraperitoneal route of vaccination. We injected the fish, and the fish this time were much larger, were about 70 grams. I would like to stress out here that Aeromonas is a pathogen, at least for sea bass, that affects mostly bigger fish. And even in this case, we had really good results, an RPS of 62.5% at the end of the challenge test. And similarly, we have even better results with Vibrio harvae. Here we followed the complete vaccination scheme. We started with immersion, and then we vaccinated the fish intraperitoneally with the Vibrio harvae, and we did the challenge with a homologous strain, and we had results, really, really good results, an RPS of 92.5%. So what I would like to tell you is that because these vaccines are made using the same pathogen that has caused the disease, they are very, very effective. Now, if we go to the regulatory issues of autogenous vaccines, as I said, regarding their use, the condition is that there is no commercial vaccine available for that particular disease. So far, regarding their production, there was no requirement at the EU level. So you could actually produce these vaccines without any special equipment or special facility. There was some regulations in several countries like Germany and the UK, but in most of the countries, autogenous vaccines were not regulated under the veterinary legislation. And in Europe, we have several small autogenous vaccine companies which are producing small patches for production animals. However, lately, we have seen that the demand for autogenous vaccines is growing, and the last years, I think that the doses which are used by autogenous products are more than the doses used from commercial products. So this has drawn the attention of the European Union, and there is a new EU regulation which is included in the new legislation for veterinary medicinal products, and this is the legislation that includes also the production of autogenous vaccines. And here in this new regulation, there is an introduction of a new concept, the concept of good manufacturing practice, GMP. Good manufacturing practice is a very well-known concept in the pharmaceutical industry. It's a set of quality assurance guidelines and standards that ensures the consistent production of safe and high-quality drugs by regulating manufacturing processes, facilities, and documentation. GMP guidelines are designed to minimize the risks and maintain the integrity of pharmaceutical products throughout their life cycle. How this is translated when it comes to application of these GMP schemes, there are several challenges. These regulations are constantly evolving and they become very, very hard to follow. There are very complicated processes that need to be employed in order to meet these regulations. The documentation is very, very extensive. You need to keep records of everything, and you need to have dedicated personnel that will track these procedures. The personnel should be trained continuously to these procedures and compliances. And even the materials that need to be used for the GMP production, the materials for the vaccine preparation, they should meet the GMP standards. The equipment that you are using should meet the same standards, and it's very, very expensive. So the costs which are involved in the application and the employment of GMP standards in vaccine production is very high. It includes costs for the infrastructures. You need to make huge investments in state-of-the-art facilities and equipment. You need to have personnel which is continuously trained, and it should be very, very skilled. You need to have a quality control system that you will follow all the time. You need to validate the processes. You need to monitor and be compliant with the new regulations. You need to upgrade the facility. All these things include extremely high costs. And I will show you just some pictures of GMP facilities with where you can produce vaccines. So you can see the personnel is dressed adequately. The equipment is specially designed for being used in these clean rooms. Now, as a recap for the whole story of the autogenous vaccines, the lack of authorized vaccines in aquaculture can be covered by autogenous products. These products, autogenous vaccines, are very, very effective. There is no requirement for authorization. You can bring them to the market without authorization. However, the production with the new regulation, which should be in place by 2027, if I'm not wrong, should be done in a GMP compliant facility. That includes a big investment from the company. Now we'll move to the second part of my presentation. I'm going to talk about alternatives to antibiotics. And specifically, I'm going to talk about phage therapy. And this is a subject that we have been working for more than 15 years in my lab, and we are among the first who have worked with phage therapy in aquaculture. Bacteriophages or phages are viruses that infect bacteria, exclusively bacteria. They are highly host-specific. Their specificity can go down to the strain level. That means that if you have a phage that infects a certain species of Vibrio, for example, it will infect only this species and it will not infect any other bacteria. Because they can target bacterial pathogens without affecting beneficial bacteria, they can be used as an alternative to antibiotics. Now our group has as a target not to use phages therapeutically, but prophylactically. And the application is mostly in the fish hatchery, as Mery said at the beginning of the introduction. Modern fish hatcheries are intensive systems where we grow fish in really high densities. These are areas of high biosecurity where we use advanced technologies for water treatment in order to produce water of the adequate water quality, but also to avoid the entrance of pathogens. And these technologies usually come with huge investments. However, despite our efforts to avoid the entrance of pathogens, we still have bacteria in the systems and in the rearing tanks. And this is because especially in the marine aquaculture and in the Mediterranean aquaculture, we have to use live feeds during the early developmental stages of the fish. And I will show you an example with gillhead cyprin, one of the most popular fish species that we grow here in the Mediterranean. So we have to use, there is a critical time window that maybe it exceeds 25 days, maybe it's more, but at least for the first 25 to 30 days, we have to use microalgae, rotifers, and artemia before we go to the weaning of the fish with the dry feeds. So at the early stages of the fish, larva rearing tanks look like a microbial soup. We have minimum water renewal. We add microalgae, rotifers, and copypods, and all these live prey, live zooplankton organisms come with their own bacteria, their own microbiome, which is transferred to the rearing water and also to the developing fish. Very recently, we finished with a project, a national Greek project, that we mapped the microbiome of 3 different Mediterranean hatcheries. We used our own hatchery because we have a commercial scale hatchery in HCMR. So the first one is the research hatchery in HCMR, and 2 commercial ones which are located in Greece. These 2 hatcheries use different sources of water, borehole and marine water directly from the sea. Anyway, what we have seen is a big diversity in the microbiome that is seasonal, because we monitor this microbiome during a whole production season. But there is also differences. There are differences between the different sections of the life feeds. There are differences between rotifers, algae, and artemia. What is really important to show you here is that I will just highlight what we have seen in the microbiome of our hatchery. And I'm focusing just in Vibrios. Vibrio is a very big genus of bacteria, which are very common in marine water, that unfortunately has several members which can become pathogenic, or they are potential pathogenic for fish. And I will highlight, because of course you cannot see it from this slide, but I will highlight here the bacteria which have the potential to create problems in the fish. And as you can see, there are many of them in the live feeds. In this table, I have included only the most important pathogenic bacteria of the genus, and you can see their quantities, their proportions in the different areas is extremely high. We have like 4% of Vibrio aquilarum in the rotifers, or 14% of Vibrio genolithicus to 16% in artemia in the commercial hatcheries. Vibrios are typically between 10 to the 3rd to 10 to the 7th CFU per ml in the live feeds, and they can become problematic when they are above 10 to the 5th. And usually this is the threshold for most of the hatchery managers to treat. And what I mean by treating live feeds is either disinfect with a disinfectant, or use antibiotics directly in the live feeds. If you do that, you destroy all the bacteria. It's a treatment that is not selective. It goes for most of the bacteria which are present. Now, our idea in our group is to use, we know that live feeds is the main gate of entrance for bacteria in the fish tanks. This can be seen as a threat if the focus is on the entrance of the pathogen, the possibility of disease outbreak, morbidity and mortality of the fish. But it also can be seen as an opportunity. If we can save, for example, a healthy microbiome, we will result in better performance later on, improved quality of the juveniles, and higher survival during grow out. So our idea is to take the phages and use them as smart disinfectant in the fish hatcheries. As I said at the beginning, phages are highly host specific, so we can target only opportunistic pathogens, leaving beneficial bacteria unaffected. And by doing so, we can control and regulate the microbiome of the developing larvae. So this is our idea, and this is our plan. In our group, we work mostly with Vibrios, as I said, and these 4 species are the main targets we have: Vibrio genolithicus, aquilarum, Harvey, and splendidus. And these are some of the phages that we have selected for this type of work. These phages are the exception. At the beginning, I said they are highly host specific, sometimes down to the strain level, but there are exceptions. You can find, not easily, but you can find phages that they have a broader host range. They can infect several strains of a given species, and sometimes they can infect different species of the same genus. And these 4 are some of the phages that are the best phages that we have isolated. They have a very, very broad host range. And I will show you some of the results using these phages in the live feeds. This is a 4-hour application of one phage against the total Vibrios of Artemia. And you can see that we can have a reduction of more than 90% of the total Vibrios in our cultures, even within 4 hours. And we have repeated this trial with a combination of different phages. We call this combination a cocktail of phages, and we have similar, even better results than the other one. And it's important to use a combination, a cocktail of phages, because if you do that, you reduce the possibility of emergence of resistance from bacteria against the phage infection. We have also tried phage therapy therapeutically in fish larvae. We are using a system for challenge which consists of a 96 well plate, ELISA plate. We inoculate every cell with a fertilized egg. We have, as I said, we have a hatchery, so we have a continuous production of Gilbert cyprin. So we put an egg in every cell, and we can make infections. So in 5 days, you can see this is the mortality of the fish that were infected with Vibrio larvae, the survival of the fish infected with Vibrio larvae. And this line is the group which was treated with the phages after the infection. So we have a much better survival than the control group. Of course, phage therapy is not without challenges. One of the biggest challenges is the variability in the opportunistic pathogens, together with the host specificity of the phages. Especially in our situation where we are working with Vibrios. Vibrios are very diverse. And if you want to be successful, you need to find phages with a broad host range. And as I said, this is not very easy to find. Another problem is with endotoxins. When you are propagating the phages, you need bacteria to grow the phages, to multiply them. These phages will lyse the bacteria, and probably they will release endotoxins in the medium. These endotoxins are very immunogenic. They are very toxic, and they should be removed, which is very expensive and very difficult to do. The biggest, however, challenge is the rapid development of resistance. Bacteria will develop resistance against phages, which will develop very fast. This resistance is different with the resistance that we see in antibiotics. It's a different mechanism. In order to avoid this resistance and to overcome the problem of resistance, you need to use multiple phages that will attack this specific strain, and preferably phages that have different receptors. And this, again, is not very easy to achieve. In aquaculture, also, you need to treat very large volumes of water. So you need to find a way to propagate the phages effectively, to have a sufficient amount. And last but not least is the licensing procedure of phage therapy products. Now here, if the claim is therapy, if you say that my product is going to be used as a treatment, it's a nightmare. You're never going to make it. There are specific requirements which are very difficult to meet regarding safety, efficacy, production in GMP facility. This is out of the question especially for aquaculture. And if you are using a cocktail, then you should do every single step for the licensing for every single component of the preparation. So licensing as a treatment, in my opinion, it's impossible. However, there is a way out. You can license these products as a biocontrol agents, as, let's say, regulators of the microbiome of the water. This is what we had in our mind, and this is why we are working towards this direction, because in the end, we want to have something that can be brought in the market. So this is an easier process. It's not easy, but it's much easier and much more feasible than having a product with a claim of therapy. Nevertheless, phages have drawn the attention of the pharma, and they are considered the next $1 trillion idea, as you can see here from the publication in Forbes. And this brings me to the last part of my talk. I'm going to talk about our company, Aquatic Biologicals. As we said at the beginning, this is the first spin-off company of the Hellenic Centre for Marine Research. And it's a company that was established in order to be small and flexible biotech company to respond promptly to the emerging health challenges of aquaculture. So in Aquatic Biologicals, we are developing and producing autogenous vaccines for the aquaculture industry. Our focus and specialty and specialization is aquaculture. We work very, very hard in the development of alternatives for antibiotics, like phage therapy products. And this is one of the highest priorities we have in the company, in our research group. But we also offer advanced diagnostic services for the aquaculture companies. We use state-of-the-art omics technology, genomics. We use reverse vaccinology in order to select the appropriate strains for our vaccines. And we also offer consulting. We work closely with the fish farms in order to make tailor-made vaccines that can offer solutions to their own problems. This is our team. You can see not only me, but Dita Tokari, which is the head in my lab. Nancy Durala is a very experienced fish veterinarian. She was working for many years in the aquaculture industry. And several people from my lab, former students, PhD students that were part in the founding team of Aquatic Biologicals. Our strategic partners is IRIDA. IRIDA is Greece's largest fish feed producer, which provides top quality nutrition for aquaculture fish. It has acquired 51% of Aquatic Biologicals and has become the major investor in our endeavors. And it offers not only the money and the expertise they have, but also network opportunities with their own clientele. And of course, Hellenic Centre for Marine Research, which is a public research organization, and this is where my lab is. And we have a very nice agreement as a company of HCMR. We can use the laboratories and the facilities in order to develop new products that will be commercialized through the company. And of course, HCMR will have a stake on this process. This is a timeline, the history of the company. We started out as a research program within HCMR. That was in 2015. By 2017, we had already developed and tested some vaccines that were tested not only in the lab, but also in field trials. In 2019, we established the company. We set up the company, Aquatic Biologicals. We had IRIDA coming in the company in 2021. 2022 was a very significant year for us because we were awarded as the best spin-off company from Elevate Greece, which is a national competition here in Greece. And we received support from the Greek state to develop our own facilities, which is now an ongoing process. We are building a new facility, and I will show you this later on in the slides. This is from the award that we received from the Ministry of Development and Investments in 2022. And this is the facilities that we are building right now in Patras. In Patras, our mother company now, IRIDA, has a very, very nice place that we are using in order to build this GMP compliant facility to produce autogenous vaccines. This picture, the last picture here, is from last month, so the progress is rapid. We expect to have the facility running running the tests, I mean, in February. We have a very strong scientific and research background, and therefore we can attract funding for R&D. We already have some projects running. We are part also of the Cure for Aqua. Cure for Aqua is the biggest EU projects for aquaculture health in Europe. We participate both as HCMR, but also as Aquatic Biologicals. And this is the list of what we have achieved all these years. We were awarded the Blue Investment Readiness Assistant Award. We were selected as within the 20 most promising startups in Europe in the Blue Invest Day in Brussels. This is again from the European Union. And we also selected as in the 10 most promising startups in aquatic animal health from the Animal Health Investment Forum that was in London. As I said, the biggest award was we named the spin-off of the year for 2022, and we have participated in several research projects that will help us develop more products and develop the company. And with this, I would like to share the website. You can visit this website and see our activities. And I have also included my email. So if you want to contact me and get in touch with me, you can use this email. And with this, I would like to thank you very much for your attention. And I'm in your disposal for any questions. Thank you very much.
Thank you very much, Pantelis, for a very interesting presentation, and congratulations on all the achievements of your company. It's very, very interesting. We have the Q&A section for all the participants. So if you want to ask something to Pantelis, you can either raise your hand or type it in the Q&A section. The first question that we have comes from an anonymous attendee who wants to know if vaccines can be used in the Mediterranean, if vaccines used in the Mediterranean can be used in other regions too.
No, this is exactly the definition of an autogenous. If our friend is asking about autogenous vaccines, this is exactly the definition of an autogenous vaccine. You should make your own vaccine. You either buy a licensed vaccine. So if you have a vaccine that covers this particular disease and it is intended for the animal that you want to use it, it's okay. You can buy it and use it. But if it's not in the market, you cannot use any autogenous product that is being used in the Mediterranean and other area. What you should do is to develop your own vaccine. That means that somebody should isolate the pathogen that caused the disease and make a vaccine for your own fish, for your own farm.
Okay, very interesting. Just before I go to the next question, still in the autogenous vaccines, I think it's a very powerful technique, especially nowadays with the antimicrobial resistance crisis and all this One Health issue in which we are all connected. And it's a nice example of precision medicine in aquaculture. But how long does it take to produce this autogenous vaccine?
You know, the first run will be very fast. So you go there. If you have the capacity and the expertise to identify the pathogen and understand that this is not covered by any commercial vaccine, then it depends on how many fish you need to vaccinate. But you can produce the vaccine depending on the pathogen. You can produce the vaccine very easily. Some bacteria grow very fast. Some bacteria grow more difficulty with higher difficulty. So that depends on the pathogen. But generally speaking, if you have an outbreak, the next season you can prepare a vaccine for that particular farm. It's very easy. What is important, however, is to work closely with the fish farm and fine-tune your vaccine, because you may have not selected the appropriate strain. You need to see how it works. So at the beginning, it could be a trial and error situation, unless you have the capacity to be very certain about the pathogenicity of the strain. In HCMR, that was one of the big advantages we have. We have access to facilities. We can test the vaccines before we suggest them for the people. We have access to students who are working. They make their PhDs on the development of vaccines, so we can do some testing. But generally speaking, an autogenous vaccine, there is no requirement of proving the efficacy. You do it. If it works, it works. If it doesn't work, you try something else. This is the law.
Okay. There is a lot of interest about phage therapy in the Q&A section. Do you expect phage therapy to be included in any laws or regulations?
Yes, there is a very big interest lately for phage therapy. There is a lobbying right now in the EU level how to legislate how to regulate their use. not only for animals, but most importantly for humans. So yes, I expect that we will have regulations. I have also already participated in some of the meetings of the EU regarding the use of phage therapy. It's not an easy task, but we are going to that direction. So I believe I'm very optimistic that in the future we will have the allowance of using phages. In the end, it's a matter of evaluating the risk. What do you want, to leave antimicrobial resistance unaddressed or do something? We need to act very, very promptly.
There is a question by Jusa-Pekka Virtanen.
Yes, I see the question. How do you deliver bacteriophages to the target? When it comes to fish, we can put them directly in the water. So that is the easiest way. But there are so many different ways of administering phages. We have also used phages in the feed. That depends. But the most common way is through the water, directly.
Okay.
Is time of administration an important factor in phage therapy? Of course it is. What it is important, phages, they need bacteria to propagate. So the phages will work very nicely if you have a lot of the bacterial target in the water. So that comes also for the time. So, and of course, it depends on every phage that you are using. And in the lab, we are evaluating these parameters. We need to address this issue. We need to know when to add the phages, how long it will take until they start to propagate autonomously, and how much time it will require to bring the titer of the bacteria, the concentration of bacteria to zero. So this is very, very important, and of course it matters. It's an important factor.
Thank you. Nicolas Baudreau, I misunderstood the part of the presentation. How can phage therapy be used prophylactically in fish eggs?
Okay, I didn't say anything about fish eggs, although we can use them in fish eggs. I said that we are using the phages in the live feed, so we disinfect, at least in my group, we use the phages prophylactically in the live feeds. We know that, and I showed you in the presentation, that the live feeds have certain bacterial species within the microbiome which can become dangerous and they can become pathogenic for fish. So phage therapy as a prophylactic treatment is meant in order to avoid the entrance of this pathogen, to reduce the concentration of the unwanted bacteria without affecting the beneficial ones. This is what I said. But you can use the eggs, you can use the phages in eggs. I would not advise it though. Most of the hatcheries usually disinfect the eggs with disinfectants. There are other people who believe that the microbiome of the fish eggs is important for the larvae, so there is a big debate about the eggs, but I didn't say anything myself.
Thanks for the clarification. Is there any risk of phage resistance developing in the future? And how many novel phages are there to be discovered with reference to the reduced number of antibiotics?
That is a very, very nice question. So this resistance against phage infection is inevitable. It will happen for sure. It's 100% certain. It's not a risk. We are not going to have any problem with the development of resistance against bacteria. There are ways to overcome this resistance. Resistance usually comes at a cost. It's not free for the bacteria to develop resistance. And what we have seen is that most of the bacteria who are resistant to phage infection, they become less virulent. But apart from that, the second part of the question was more interesting. Are there many phages? Actually, the number of phages is astonishing. They have estimated that the total number of phages in the planet is 10 to the 31st. This is an astronomical number. This is a huge number. So there are very, very huge reservoir of unexplored phages. So you can do that for your whole research career. You can isolate and characterize phages continuously. You are never out of phages. And you can actually train the phages. You can develop, you can make the phages overcome the resistance. You can make them adapt to the resistant mutants. So there are ways of doing it.
Thank you very much. So somebody is asking how to access your research services.
How to?
To access the research services you provide.
You can email me and we can get in touch and we can discuss about specific situations.
Okay. I will add to your answer that we have also transnational access calls in which maybe your facilities are
Yes,
available.
we can. We also participate. We participate as a research center in EMBRC, so we can host, and we usually host people from academic institutes, but also from companies. But we're also part of the Aqua Excel platform. So if you have researchers that they want to access our facilities, we can discuss about this. There are options of collaboration.
Okay. Can you say something about how to produce the phages?
Yes, this is a question that it's not so easy to respond. In my lab, we are using bioreactors for phage propagation, and we can go up to 10 to the 12th PFU per ml. So very, very concentrated phages we can produce very efficacious. The problem is not the production. We can produce phages very easily. The problem is how to get rid of the unwanted proteins, which are the remnants of the bacterial lysate. This is the difficult part. We centrifuge, we separate the two fragments, we can separate the phages. Nevertheless, there are proteins which remain in the final product, which require further process, which is very expensive. That is the problem, not the production. And phages are very stable. You can keep them in the refrigerator and they remain stable for many years. So they are very, very fine.
Okay. For the next one, there is Ainoub Bounasir from Morocco, and I would like to give him the floor because he can maybe share with us the problem that he has found with Vibrio harveyi. I don't know, Christine, if you can give us a hand. So Ainoub, I don't know if you can open your microphone. Okay, Ayuba is not there.
Anyway, the Vibrio harveyi is very problematic. I understand the question by Ayub, and it's very problematic not only in Morocco, everywhere, because it's a very persistent pathogen. You can treat it with antibiotics, and then you have a recurrent infection, and again and again and again, until the temperature goes below 20 degrees or 19 degrees. So it's a continuous problem. It's not easily handled. I think vaccination is a very good step because most of the species are very susceptible, usually the first summer of their life in the cages when they are exposed. So fish of like 50 to 60 grams, these are the most sensitive part of the population. So if you can vaccinate the fish and overcome or reduce the impact of outbreak during the first year, the first summer, I think the second summer would be much easier. This is my personal opinion. Of course, it changes from area to area. And I assume in Morocco, they have really high temperatures, especially in the Mediterranean part of Morocco. So if it's really high temperature throughout the year, probably they will have problems throughout the year. In the west part of Morocco, which is in Atlantic, I think the situation would be a little bit better.
Great. I also had this question. Can these therapies be used for other aquaculture species? Like
yes, phage therapy can be used for any type of you are treating a bacterial pathogen. So if whatever you have in your facility, you can treat it with the phages. But I need to be very clear here. This is still experimental. We haven't reached to the point where we will have a product that will address every single disease. So this is specific to, we need to see what is the exact problem. And usually we have phages for most of the pathogens, but not for all. For example, we have been working for years in order to find phages against Photobacterium Damsella piscicida, what we call Pasteurella in aquaculture. We never had a single phage that could target this bacteria, and we tried hard.
And for the vaccines, is the same? We can use it in other aquaculture species?
With the vaccines, yes, again, but autogenous vaccine is an empirical product. So it could sound very easy from our presentation. It's not. You need to know the immune system of the fish. You need to know when to vaccinate, how to vaccinate. It's not so easy. So there are several aspects that they need to be involved. I didn't have the time to expand my presentation to all aspects. It could sound easier than it is, but yes, vaccination can address almost all fish species which are being cultured.
Okay.
As far as they have an immune system that can develop antibodies, vaccination is this. You train the organism to develop antibodies that can be used when it encounters the pathogen. So,
for protection. So coming back to phage therapy, how is risk of horizontal gene transfer?
yeah, it's a big risk. It's a very, very valid question. And I assume they are talking about phage therapy, not gene therapy. So this is a very valid question. And this is something that I didn't say in my presentation. In phage therapy, we are using exclusively lytic phages. There are two types of phages, lytic and temperate. Temperate phage can insert their genome within the chromosome of the host bacterium. If they do so, the genes which are part of the phages can become genes of the host. So they can offer new attributes, new characteristics to the bacterial host. And this is something that we do not want. A very good example is cholera. Vibrio cholera, the pathogen which is causing the disease cholera. The toxin, the major toxin of cholera is within an area of a phage. So it's a toxin which is encoded in a prophage. Prophage is called the phage which is inserted in the chromosome of the bacteria from a temperate phage, and this can be transferred horizontally. How do we exclude temperate phage with genomics? Nowadays, we analyze all the phages that we are using. We run the sequence of the host of the phage, and we look very, very carefully for genes which are signature genes for temperateness, for this ability to be integrated within the chromosome. And we can exclude the phages very safely, the phages that have this attribute of transferring genes from one species to the other. But it's a very valid question.
I have another question about this phage therapy, and I wanted to know if you have done a study on how does it affect the microbiome once you take out these infectious species?
Yeah, that is actually what we are doing in the Cure for Aqua. I mentioned Cure for Aqua. Cure for Aqua is the biggest research project right now in Europe for aquaculture animals, for aquaculture health. And within Cure for Aqua, actually, we are running the experiment right now. We are treating live feeds with phages, but also probiotics. We try to regulate the microbiome, and we follow what will happen to the fish later on when they are in the grow out phase in the cages. So the question here is your question. What will happen if we intervene in the microbiome of the fish at the early stages? Is this going to be beneficial or not? We don't know. I cannot answer this question. What we know for certainty is that microbiome is very, very important for the developing fish. And it's also connected to the first establishment. So the very early stages, the fish larvae, the establishment of a microbiome at these stages probably is extremely significant. And this is a question that we will answer in Cure for Aqua, hopefully. And we have very good partners involved. We have the Functional Genomics Center in Zurich, who is doing this genomic analysis and gene expression. We have the epigenetic effect of these treatments. So there are many aspects of research questions which are involved, but most importantly, what will be the effect of this treatment early on, later during the fish are already grown, several months after the treatment.
Very much looking forward for the answer.
We also
to ask you something that is not related to research, because you are an entrepreneur too. So I wanted to know if you could share with us how has it been this new career path of being an entrepreneur for you?
Yeah, it was very interesting since the beginning. It's not easy, especially in countries like Greece. In Greece, we don't have this experience in creating businesses. especially if you are starting from the public sector. My research center is a public research institute, so it was not easy to start. We didn't have any regulation, any roadmap. We made the roadmap ourselves. So I had to discuss with my administration. I had to explain what is the reason behind the need to make a company, because we had a product. We had not only one, but we had products in the lab, but we knew that we could not use these products unless we have a GMP facility. So this GMP requirement was actually the driving force for us to establish the company. You cannot invest like 1 or 2 million euros within a public research center. It's impossible. You need to have a company. It's not easy. We were very lucky, however, in our case, because we attracted the interest of IRIDA. IRIDA, which is the company who has invested the money in our business, is a very serious feed company which has great knowledge of the sector. They are highly respected by the Greek aquaculture companies, and it's very, very helpful to have people with expertise and knowledge in this field, in this particular field. It's not the same if you have an investor from, let's say, a fund. It's completely different. So in our situation, I think the best thing that happened was that we attracted the interest of companies which are related to what we are trying to do.
Okay,
but it's not easy. It was fun at the beginning. So far it was fun. Now it's not fun because there are so many difficulties. You are working with so much money which are involved in the investment, and since it's not my money, this is much harder for me. If it was my money, I wouldn't be so worried. This is my honest response on that.
Yeah, I hear you. I hear you. Especially once when you are a researcher, you lack of certain skills that you start gathering on the way, and yeah.
And right now, I'm serving both. I have two hats, as we say. I'm a researcher and an entrepreneur.
Yes.
So it's not easy.
Very courageous. We have still 2 questions in the Q&A, and I don't want to close the webinar before answering them. So what about creating a temperate phage that provides the bacterium with a detrimental feature?
I think this is what I said. We do not use temperate phages. When we isolate phages and characterize phages, the first thing that we are trying to do is to exclude temperate phage. So this is something that it can happen if you are not careful, but if you know how to do it, you can easily exclude these phages. So no, we don't use temperate phages. And the next question is that, is the treatment of bacterial infections in aquaculture, particularly in open systems such as floating cages, effective? No, it's not. It would work much better in a closed system, in a recirculating system, in a static system, in tanks. It's much better. When it comes to floating cages, what we can do, and this is a proposal that we have already submitted, and if we are lucky, we can get funding, we will follow it, to use phages in the feeds and to use them as functional feeds in order to reduce the probability of having an outbreak of, let's say, Vibrio larvae, because Vibrio larvae causes enteritis, it goes through the gut, and if we can target this organ with phages in feeds, then we can have a system, we can have an approach that can be applied in floating cages as well.
Okay. Well, we don't have any more questions. So I would just like to thank you again, Pantelis, for joining us in this webinar. It was very interesting, your presentation, and we wish you all the luck for your new research projects and the future of your company. I would just like to let know our participants that your webinar and all the past technology webinars, well, only one past technology webinar about aquaculture, are available in our YouTube channel. And also that our next series of technology webinars will be on marine observation. So check out and follow us, our news, so you can follow our next webinar. Thank you again, Pantelis, and thank you to all my team,
Thank you.
EMBRC team who are behind, Gwenael Walter in the communications, Paulina Ramirez, Estela Alexandorf, and Cristina Pavludi in the back end of this webinar. Thank you very much for everyone for being here. Have a nice afternoon, and we leave a little poll open if you want to share with us how did you like the webinar, and also if you have other related topics that you would like us to discuss in the future webinars. Thank you very much. Bye.